那我们 OK。好，各位同学，我们演讲开始哈。就是今天很高兴呢，请到这个中研院分身所林伟翔博士，他现在是中研院助研究员。他虽然说是中研院分身所，但他数学很强很强，我可以盖章保证哈。那他今天要跟我们讲一个很棒的主题，就是所谓的 Biomass Transfer Algorithm Relation Day Work。然后它这边有个叫 differential equation 的 approach。那其实这个是很新的研究。那他会给我们看很多现象，然后跟我们讲怎么样数学来去研究这个。那因为今天呢，我们有这个韩国老师在这边，所以我们待会会用英文给演讲，可以哈。那其实未来如果有一些国外的演讲者也会讲英文啊，其实你们硕士班从现在开始就要习惯用英文听演讲，但是你们问问题可以用中文问，可以吗？用日语问可能也可以吧，对不对？如果要的话。好，没有德语。好 ，OK， 好，有问题吗？好，那没有的话，我们就欢迎这个林伟翔博士。
好，谢谢家园今天很高兴 I'm very happy today to share my work with you guys. So I'm working on mathematical biology and biophysics model. So today I'm going to talk about growth of the system. So let me slowly start with some. So if you study biology, like a cell system or ecology, or in an economy system, their common behavior is they are able to grow and expand. So if you grow a cell, they can double and become more cells. And if you have an ecosystem, and you can establish a food web, and this food web can expand and create more individual in the ecosystem. Or you can have an economic system. Economic system, you can also have economic growth rate, right? Like a GDP, how much percent you grow per year. So all this system is able to expand. And my research interest is to study how to have a model to describe expanding system. And the reason can be very theoretical, but also practical. For example, in the cancer research, you want to stop the growth of the cancer. So you need to find a drug to suppress the growth. But first you need to know why cancer can have a model for cancer to grow. And sometimes in agriculture, you want something grow faster, like rice. So you also want to have a general model to describe growth phenomena. So growth phenomena in physical science or biological science is relative open question. And the reason is, in physics, most of the system you learn in high school is static equilibrium. And some of them are steady state, which means the influx and efflux will cancel out each other, but they are not the same. But actually, when you have a growing system, you now need to consider influx need to be bigger than efflux. Otherwise, it will never accumulate things. So every time you need to have a slightly bigger influx than efflux, and the system can grow. And finally, in biology, the traditional model is to assume called balanced growth. Means everything in this system is proportional. Like when you grow, for example, your head grow 5%, then your body grow 5%. Everything is proportional. But as we will see, in nature, sometimes it's not proportional. Sometimes this proportion can still oscillation. So I'll quickly go to some of the example in physics. For example, the crystal is a static equilibrium because you can have crystal in solution or in crystal form. Steady state, one of the example is cytoskeleton. I don't know if anyone knows what is actin filament, but it's a very important cytoskeleton in the cell. That if you want to maintain a cytoskeleton like in a muscle, it can grow in one side and then shrink from the other side. But in general, the length is still keep a homeostasis. So for the growth model, there are many models to explain cell growth. But so far, these models are not as generalized as a framework. Each type of cell, for example, they will make a new model. And finally, we can have a more complex growing behavior. So let me show you a movie. This is culture chamber, and the cell you see here are E. coli. So these E. coli cells, you can see they are growing, so they push more E. coli out. And the color here demonstrates how much energy molecule there are in the cell. If you see a purple cell, which means it has a lot of energy molecule. If you see green one, it has fewer energy molecule. And definitely when they're growing, they are not in a stable ratio. Their concentration of ATP can change. So this is like a motivation. In mathematics, we need model to describe things both growing and dynamics. So of course, growing systems are still very complicated. We want to focus, in this study, we want to focus on exponential growth. And the reason is exponential growth has been observed in many systems. This is a simple chemograph that they image this cell by 20 times, different time points. And the cell will gradually elongate. But this elongation is not linear. It's actually more like an exponential curve. And another study is from my colleagues that he blocked cell division. So the cell can grow from one to a very long one. And you can see that in the long term, the cell length, cell area is proportional with the growth absolute area. So this is like also indicate exponential growth. So exponential growth, at least in microbiology, is very common. But exponential growth, of course, everyone knows linear system will have exponential growth. But actually, all these systems are nonlinear systems. It's not trivial why they can grow exponentially. So what I want to do is a mathematical setting, like more general, how to generate exponential growing system. So the mathematical structure we use is called a reaction network. In this case, we have an environment which has unlimited resource and a system. So in a system, you can have reactions which convert different, you can think metabolite into another metabolite. Okay. So this system, the type of every node is the same, but the system can get bigger and bigger. For example, in the time t0, you can have 1,000 molecular here, and after 10 minutes, it can be 2,000. So this system can expand. So our mathematical model is to use ODE in this case. So as you can imagine, each node, you can have influx and efflux. So the change of amount of molecule in this node is basically influx minus efflux. So the influx and efflux are related by this storage geometry matrix. And this is like a very well-studied structure in mathematics. So now we have influx and efflux as a J, as a flux function. But we haven't specified what is J. Okay. So the reality is, if you use arbitrary J function, then the system doesn't need to have exponential growth. You can have linear growth, you can have finite time blow up, or you can have even other things, non-biological. For example, each molecule here should be non-negative. But if you choose some constant efflux, then the system will have negative things, so which means it's not a good model. So our next question is, what kind of flux is good to generate exponential growth? So what we want to find is a class of flux function. So, okay, so I need to rigorously define first what is exponential growth. So in our case, we call system size N is the summation of all amount, total amount. And then we will call the rescale biomass by Y, so which means if you have a very big system, and then this total amount, you can think this is N, and then you have certain Xi. So Y is basically normalize this pie chart by N. So Y is a number between 0 and 1. You can think this is a rescale or relative biomass. And what we think exponential growth means, this terms lambda is a limit of log nt divided by t. If this limit converge, we say it has exponential growth rate lambda. So exponential growth doesn't need to be exactly exponential curve. As long as this index converge, we call it, we count it as exponential growth. Okay, and this also equal to the long-term average of this instantaneous growth rate. So dn over dt is the absolute growth rate, and if you normalize to the system size, it's an instantaneous growth rate. And if you do a time average in the long term, and this average exists, then this will also equal to what we call the long-term growth rate. So now, we go back to the question, what kind of flux function J will give us exponential growth? So what I found is there's a special class of flux function we call the scalable flux. And in mathematics, it's usually called homogeneous of degree 1. So homogeneous degree 1 is a group of functions, including linear function. For example, if you have this j equal to x1 plus 10x2, then if you increase x1 and x2 both by 2 times, the flux will increase by 2 times. So this is homogeneous, and degree 1 means this c outside is order is to the 1 power. Okay, but homogeneous degree 1 can also have many nonlinear functions. For example, this is a simple x1 x2 divided by this, and if you times every x by 2 times, then the flux is also 2 times, and you can even have non-monotonic nonlinear function, which is homogeneous degree 1. Okay, in functional space, if you have Rn space, homogeneous degree 1 function is actually as much as Rn minus 1. So it has so many functions can satisfy this condition. So this is a large class of functions, and what I'm going to show is if we use this to construct biological system or to model biological system, many of them will naturally go to exponential growth. And I will show you why they become exponential growth. Another relation with this homogeneous degree 1 function is it has been used in economy. So as we see, economy growth is a very long-standing question. There's a Nobel Prize Solow-Swan model, and there's a special name of this Cope-Douglas function. It's actually a simple homogeneous degree 1 function. And they also study the exponential growth of y, but in their case, y is just a scalar. So it's not a very complex ODE. In our case, it's a very complex ODE with arbitrary dimension. Okay, so now let me do a little bit slowly here to show why homogeneous degree 1 has a special property. First is this dx over dt equal to f is our nonlinear ODE. And now let's consider the relative fraction, which is x divided by n. So why? We can write down the ODE, dn over dt and dy over dt. This is just calculus. So up to now, we're just doing change of variable. We didn't do anything. This nothing new. But now, if you have homogeneous of degree 1 or scalable condition, then f of x you can rewrite as n times f of y. And remember, this doesn't need to be true. Only for the homogeneous degree 1, this will be true. And this is actually a functional relation. Because remember, this f is a vector function. So if you can have this, then the ODE here, you can pull out this n out. So dn over dt is n times this sum over y, fy. And dy over dt is this f of y minus this term. And this term, y over n dn over dt is actually a function of y only. So you can, using this scalable condition, you actually decoupled n-dimension ODE into two subsystems. One is n minus 1 dimension on the unit simplex. The other is 1 dimension on the positive axis. So of course, 1 plus n minus 1 is n. So we didn't lose anything. This is the dynamics. So now what is the good thing to do this coordinate transformation is first, this 1 dimension talk about is the long-term growth rate, is the axis for long-term growth rate. And this n minus 1 dimension now is in a bounded simplex space. So the simplex space, what I mean is a space that all the components add up equal to 1. So in 2D, simple space will be a line, segment. In 3D, it will be a triangle, and in higher D will be like a pyramid. So the good thing is, okay, once we decompose this to n direct sum of y, we can calculate the ODE into two subsystems. So the simplex dynamics can do long-term average, and the radial dynamics we can calculate long-term growth rate. And there's no more tricks here. I'll just quickly go through the summary that for the scalable condition, the long-term growth rate is equal to the long-term average of the mu t. Mu t is the short-term growth rate. And if this trajectory in the simplex, your trajectory go to some attractor, which you can average on this attractor, we call ergodic condition, then your long-term growth rate exists. So this is existence theory. Okay. So let me give quick example. So this is a replacer model. And here we have flux homogeneous of degree 1, 4 flux, and we want to see what is the growth rate for this system. And for different parameters, sometimes you will get balanced growth. So here we see the y-axis in log scale. So if you see a line, which means this is actually exponential growth because this is log scale. And here you have oscillation in the long term. Okay, so this is actually non-balanced growth, but in the long term, it still converges to an exponential growth rate. So our conclusion, first part, is using the function homogeneous degree 1, we can construct a family of ODE, which all of them has exponential growth. And in biology, this is very interesting already, because when we write down an equation for the cell, it's rarely complete. It's difficult to know every component in the cell. So usually you want the entire framework that is flexible. Sometimes you can include more things, sometimes you take out things to make it simpler. But you still want the system to have exponential growth. So you can compare this exponential growth rate. I want to make one more point. In evolution biology, sometimes we talk about fitness, right? So the cell will divide faster, will be more competitive in the ecosystem. And ecosystem, if you give them enough resource, fitness is almost exponential growth rate. So this is also a model for evolution. You can have a reaction network. This is simple, but you can also have a very complex reaction network, something like this. And then you can still try to calculate, okay, if I add one more function here, will it improve its fitness? And that can have some potential application in engineering, because sometimes in engineering, we can do bioengineering to change this function. And then we'll try to see if we can make the cell grow faster. Okay, so far, is there any question? And because then I'm going to a second part, which is about how to convert this framework into a DDE, because so far we talk about ODE.
I have a question. I'm not sure whether this is a good question. Are there any first principle to guarantee the scalable function, or it's just a
Yes. Yes. Okay, this is a wonderful question. I actually write in the paper. So in biochemistry, if you assume that the cell volume, usually what you see is cell has certain DNA or protein, but also has a total volume, right? If you assume this cell volume is proportional to the biomass, and then you assume every flux function is a function of concentration, these are two assumptions, then you will have scalable. Because when you grow in volume, you pick a unit volume, the concentration here will give you scalable flux. So the question is, now, does this assumption reasonable? Because sometimes when your cell get bigger, your volume doesn't need to be scaled with power 1. You can scale in general with certain power alpha, right? So if alpha equal to 1, then we show this is scalable. So in certain linear growth system, it's scalable. Or well mixed is scalable. Yeah. Okay. So if you have a question, you can also discuss later. So now, I just go through the basic property for homogeneous function of degree 1. So now I want to go to the next part is, can we analyze this ODE? as a reaction network using a DDE. And then this ODE and DDE has the same long-term growth rate. So why do we need to do this? It will be apparent in 10 minutes, but let me quickly say, n-dimensional ODE or a roughly one-dimensional DDE, I think this is easier than this, right? So this is already a motivation that if I only care about the long-term growth rate.
I have to make a remark. Okay?
Yes.
There are n equation, n ODE, a scalar equation of
Yeah.
DDE, but DDE is infinite dimensional. Be careful, okay?
Oh, okay, yeah, okay. One dimension, okay, okay.
If you just count the number of equations, yes, it should be count the dimension.
Dimensionality, yeah. Okay, I agree, yes. Okay, thank you for the remark. So how do we, okay, so what I'm going to show you is a scheme that for scalable ODE, we will have a systematic way to construct DDE such that every ODE will have one DDE. Okay. And this one DDE has the same long-term growth rate as this ODE. So to solve the long-term growth rate or to understand the long-term growth rate, sometimes we can do here, but sometimes we can do with the DDE setting. And sometimes DDE will be more intuitive. So let me start slowly. So this is what, let's go back to the physical meaning of our system. Physically, reaction network usually has mass conservation, which means if you have mass come in certain, like a fluid dynamics, if you have mass come in this position and you have mass go out, then the total mass here is influx subtract efflux. Okay, you cannot destroy the mass in this physical setting, and in our biological setting, it's the same. So in mass conservation system, fluid dynamics can have two perspectives. One is called Eulerian, which means you fix an absolute position and you calculate influx minus efflux. You get a change rate of here. Or you can use another perspective called Lagrangian. It's like you track a group of mass, like a group of atom or group of fluid. And then in the space, once you track them, you can write down another kind of equation, usually related with co-derivative. So this is another equation here, but you can also have same solution both on Eulerian or Lagrangian. And the only thing different is Eulerian is an absolute coordinate. Lagrangian you track with the biomass moving. So in reaction network, we have the same analogy. The system ODE you see is you look at each node and you do influx minus efflux. But you can also think, since biomass, you cannot destroy it, you can discretize the biomass conceptually. And once you discretize, it's no longer ODE, it will become a Markov process. And this Markov process, because mass conservation, you can track this mass unit on the reaction network, like tracking a Markov on a Markov graph. So what I'm going to show you is using this, I was able to rewrite the long-term trend of ODE into DDE. Okay, so the first thing, let's observe some intuition. So the intuition is, if you eat something from the environment, E, into the system, then this flux, what we call is boundary influx, labeled as red one. So if the cell need to grow, boundary influx need to be at least positive. And then you need to think about, they can also leave the system. So there's also boundary efflux, labeled as purple. So the total growth rate is basically boundary influx minus boundary efflux divided by system size. And this, if this is always positive, then your system will keep growing. But it doesn't need to be always positive. As long as your long-term average is positive, the system can grow. But does this mean we can solve the ODE easier? And the answer is probably not, because the reason is, okay. The reason is, okay, let's assume this boundary influx and efflux, they are all scalable flux. The problem is, wait, let me see. Okay. The problem is, in order to know the boundary influx, this flux function can depend on things inside the system. So although here we only have 3 functions instead of many functions, these 3 functions still depends on n-dimension variable. It's not only 3 variable. So this doesn't help. But what can we help is we can track gatekeeper growth. So this is probably a relative new concept. What we call gatekeeper is, if we look at node which promote this flux, then they are called gatekeeper node. In this case, there are 3 boundary influx. So we assume there are 3 nodes, probably these 3 is essential for this influx. Okay, this is purely a mathematical assumption. In biology, it doesn't need to have always like this. But usually in a cell, if you want to grow, you have transporters for this nutrient. For example, carbon source, like glucose, you have glucose transporter. Nitrogen, you have nitrogen transporter. So it's very nature that each influx, there's a transporter inside. That transporter we usually call the gatekeeper here. So you can think gatekeeper collectively, they amplify themselves as an autocatalytic system. They keep moving more flux, more material inside the system. Once the material goes into the system, they, in the neuration network, eventually they make more gatekeeper. And this is like auto-positive feedback. So in a Markov process, you can think this is a process that outside, biomass outside in the environment gradually become internalized into the system. Okay, so how do we write this qualitative concept into mathematical model is the next question. But I hope everyone can get slightly the intuition is we want to track each biomass, how does it get into the system, and then after a certain time, you will reach the gatekeeper, and the gatekeeper again come back to promote more mass. It's like closing the positive feedback loop. So to illustrate the idea in rigorous mathematics, let me use a simple ecology model. So this is a simple model, what we call the simple autocatalytic pathway. In this model, you have n species in the system, and each species can convert into the next one. Okay, and once you convert into xn species, this one doesn't degrade. Instead, this one come to promote more biomass enter here. So you can think this is like a way xn catalyze itself, but this catalyzation takes time because when you create x1, it takes certain time to go back to xn. So what is the long-term growth rate for this system? Well, you can write down this is completely linear, so you write down the matrix and you calculate the top eigenvalue of the matrix. Okay, so this is a Lagrangian view. But now, as I said, you can also do Lagrangian view if you assume mass conservation. So the good thing is, if mass conservation, this model is equivalent to an ecology model like, okay, xn is like an adult insect. An adult insect will generate several eggs with rate b. And then this eggs will mature given certain time delay. After this time delay, maybe this is like 2 months, they will be more adult, generate more eggs. And this is like a positive feedback. And this positive feedback, of course, we can write as a DDE as following. So to know the time delay, we can investigate what is the time delay from the zeroth flux, which is this influx, to the last flux. So here J0 is the first flux, and Jn-1 is the last flux. So if I have labeled certain things, after maybe 2 months, it will become mature and become Jn-1. So there's a Markov relation between Jn-1 and J0. And there's a delay time distribution here. So in a linear system, this is, okay. And this, the last flux, Jn-1, is actually dxn over dt. And the first flux, because we assume this xn will promote the first flux, is xn. So this Markov relation, because our assumption will become a DDE because this is dx over dt and this is dx. Okay, and this is, I think, the only trick in this project is, this is just a normal Markov relation. But because the structure, we are not picking random node. We pick one node which can auto-catalyze itself. Then we can write down this DDE. So in this case, the delay time, because this is a linear system, we have exponential waiting time. I think everyone here probably know that if you have a first order reaction, the waiting time has a exponential distribution. Is that common or it's okay? I think in physics it's quite common, but you can think like a radioisotope decay. The time you look at the radioisotope to decay, it can be 1 minute, 5 minutes, 10 minutes. But if the average, let's say, is 10 minutes, then the distribution is an exponential distribution with average 10 minutes. And this is like, can be proved, this is memoryless in the stochastic process. So using this well-known function, because each step, you have an exponential waiting time distribution, then the total waiting time is the convolution of this. And this convolution gives you another distribution. Here I called alpha. So this alpha, I call it H. So this H is the waiting distribution. And I further times a prefactor, which is how much they amplify themselves, is B. So B times H is a new function we call catalytic kernel. This is basically, if I have 1 unit of Xn, it will amplify B times to generate many X1, and then they will become Xn later. And this amplification process has a kernel alpha tau. And by doing simple conversion, you know that the long-term growth rate follow this DDE. And the kernel here is calculated by the waiting time distribution. And if you do slightly more calculation on the Laplace domain, on the frequency domain, using Laplace transform, you can prove this relation that the Laplace transform is, which is a catalytic spectrum, satisfy this lambda equal to a lambda. And we'll come back to this. This gives us some geometry interpretation of this spectrum. So let me show you an explicit example. Suppose I have a system which X1 immediately feedback itself. In this case, amplification is B, and the waiting time is a delta function because it doesn't need to wait. It immediately feedback. Okay, after this, okay, so in this case, the catalytic spectrum is a constant spectrum. If you have 2 steps, then you need to wait this X1 convert into X2. So the spectrum is a hyperbolic decay. Okay. And if you have 2 steps, X1 equal to X2 and X2 equal to X3, then you get another waiting time and the decay is even faster. So overall, this spectrum here will intersect this diagonal with their long-term growth rate. So in this case, you can actually see, okay, this blue spectrum intersect here. So lambda 1 must be bigger than lambda 2, must be bigger than lambda 3 if you keep adding more steps. And this is intuitive, like if you want to grow fast, you want a lot of amplification and you want short waiting time. If you wait very long, of course it takes very long time to catalyze itself. Okay. So how this help us to calculate long-term growth rate? Because we can still do Eulerian, so we can actually, I say numerically, this is easiest, right? You just write down the matrix, you just calculate top eigenvalue. This is, you don't need much brain. I mean, you can program this and then calculate. While the Lagrangian is like, okay, you have some biological argument, but you get the same answer. So what's the advantage? So let me show you a simple an analog, like in the future, what we want to do in mathematical biology. In mathematical biology, as we see, we rarely can write down the full specification of the system. So sometimes we need to consider coarse graining. So for example, a cell can have 100 or 1,000 protein, but you don't want to write a very complex ODE. You classify the protein into different types, and then you only work on maybe 5 or 10 types. So coarse graining is important. So you all learn in high school. In high school, you can coarse grain a complex electric circuit into an equivalent electric circuit. And then you can predict equivalent R, which is the equivalent resistance, using the well-known formula you have in series or in parallel, right? So it's like 串联 and 并联. Okay, so you have this formula. Now, our question is, can you do similar formula with reaction network growing system? So, can we coarse grain this into a simple question? And because we have this framework, I can tell you this is impossible. And the reason is the waiting time for this system cannot be always find another waiting time of this one step. I cannot find this as this. So which means in electric circuit, because the current doesn't grow exponentially, you can find equivalent system, but in the reaction network you cannot. But we can still do something. It's not the end of the story. We can find two simple systems, okay, as an upper bound and lower bound for a more complex autocatalytic pathway. In this case, I prove that, okay, you can use a simple one with like a harmonic mean of the reaction, or you can find another one as a lower bound using this the spectrum I mentioned before. And then once you calculate this, it will guarantee that your growth rate of the middle one, the real model you work on, is bounded between these two growth rates. Okay. And the reason it can bound it because we know the spectrum. Because the upper bound spectrum is always higher than the black spectrum, which is the real one. And the black one is higher than the orange one in this region. So your long-term growth rate geometrically is bounded between these two things. And I think this is a new technique. Because if you only do the matrix eigenvalue, it's very difficult to coarse grain an n-dimensional matrix into a two-dimensional matrix and compare their top eigenvalue. This is, I think, you can try doing this, but it won't be intuitive. While here, it's much more intuitive. We just need to construct a proper system that can bound the spectrum, and then you get this very good approximation for coarse graining. So let me wrap up. The second part is, here I use a simple example, which is a line, like a single pathway, to show you I can calculate long-term growth rate with two methods. One is the traditional eigenvalue problem. The other is using the delay time. And the delay time can actually allow us to do some coarse graining approach. I'm wondering if anyone has a question so far I can discuss. Yes. So in this reaction network, because mass conservation, thank you for the question. Because of the mass conservation, the diagonal is always negative, and the off diagonal is always positive. So there's a Perron-Frobenius theory that can guarantee that this top eigenvalue is the real eigenvalue, and this is lambda. Yeah. Okay. So in the last 10 minutes, I would like to generalize, because this is one example, right? This is a very simplified reaction network. What I really need to show you is this, right? In the beginning, as I promised, how to convert every scalable one into a DDE. So, or in a more precise way, if you give me one ODE like this form, how can I write alpha and beta and gamma as a function for this ODE? And of course, this may not be easy, but this alpha, beta, gamma have its own biophysical meaning because they represent the catalytic feedback, how long you need to wait. And this beta represent the overall decay of the gatekeeper's nodes. So to do this, we need several assumptions. First is we assume as a Markov process, every flux function you can express as RA of y and xk. Okay, if so, which means if you have an upstream node xk, you can probably have other upstream node. Okay, and they will go to certain downstream. This is like a very general reaction R. So reaction rate is a function of relative fraction. It's not only function of x, but also function of y. It's a relative fraction. And then this reaction speed is proportional to x times certain nonlinear flux here, function of y. So here, this is our assumption. And second assumption is, if we have a boundary influx, we assume it's proportional with one of the node in the interior. And then we assume, okay, the trajectory will converge in the simplex. Okay, and then we assume growth rate is positive. And finally, we assume mass conservation. Okay, so these are the mathematically we need this because otherwise the theory will be not rigorous. Right? So if with this, I will first define what do we mean gatekeeper. Gatekeeper is, for example, you have 3 influx here. Suppose they depends on 3 of the node. Then these 3 nodes, I just sum them. I call this Z as a gatekeeper biomass. And the main theory is this Z satisfy a DDE in the long term if the assumption in the previous slide is hold. So not only this, I want to show you how can we construct explicitly how this alpha and beta is constructed, okay, and how we get this similar geometric equation. So the first thing is we need to use some language in Markov process because this is, we discretize the system and we think it's a Markov process. So the first step, we need to find the first heating pathway in the system. So for first heating pathway, we means like we have collection. Let's say this is a collection of node A and this is another collection of node B. First heating means a pathway you leave A and then go to somewhere else and then you hit B. Then this is a pathway, what we call the first heating. So now you can consider, yes. Okay, Z cannot directly change back to X, but the long-term growth rate of Z is the long-term growth rate of total X. So to know the long-term growth rate, we only need to focus on Z. We don't need to focus on other node. And this is there's a proposition in here. Yeah. So you can think if the system we assume has a positive long-term growth rate, this flux must be grow as fast as the total system. If this flux is slower, then the system will stop growing. Okay. So there's a proposition that by this assumption, the Z, which is the sum over the gatekeeper node, will have the same long-term growth rate as the entire system. Okay, so now we categorize reaction pathway in the system into 3 categories. One is first heating from environment to gatekeeper nodes, which is the component of Z. Okay, so for example, you have biomass here, and then you wait several times, and you hit first time hitting G, which is a collection of gatekeeper. And then the second category is like a recycle from gatekeeper to gatekeeper node. So first heating from G to G. And then the last first heating is any other nodes first heating this G. So we can prove that these 3 processes are orthogonal to each other in terms of Markov process. For example, this is like if you have a reversible pathway, here your gatekeeper is the last node X3. So the first heating pathway, actually there are infinite pathways. You can first come X1 and then go to certain reversible cycle and hit X3. So you have many first heating pathways. Each one has different probability. Okay, and category group is first heating from G to G. Is you start with X3 and you go to other node with certain time and go back to first heating back S3. This also has an infinite pathway with different geometric decay of probability. So once we have first heating pathway, we can work on the total amount of first heating, because a biomass, if it's mass conservation, you must choose one pathway to heat something. You cannot choose both. Okay, this is like mass conservation. You either choose one or you choose another one. So basically the arrival function will be summation of different pathways for this first heating process. And each process now, because this can be a nonlinear function, so this is no longer not simply exponential decay waiting time. This waiting time can be more complex. And then to know the total waiting time, you use the Sharp-Mann-Kolmogorov equation to do the Markov process. And finally, you sum up all the relevant pathway. Okay, this can be proved that these are exactly all the pathway you need to sum. And then for each pathway, you calculate amplification rate and arrival function. You will get this alpha. Okay, so I already showed you how to construct alpha from a general ODE based on the assumption we have. And for the beta, which is the general degradation of the gatekeeper, it's also there's a formula for this. It looks complicated, but actually this is just weighted how much for each gatekeeper they leave the set of G. So there's also a relative explicit formula for beta. So now once we have alpha and beta, we need to perform suitable average. Remember that in a simplex, okay, in linear system, you will go to a constant vector. So the fixed point is the attractor is 1 point. But for the simplex, you can go to limit cycle or go to other things. So you can do this, the suitable average is the ergodic average for alpha and beta. And it's proof that if you use this suitable average, okay, on the simplex, you will get a similar DDE here. So finally, I summarize. For general ODE, of course, we have a list of assumptions. But if the system satisfies this list of assumptions, then we can write down catalytic kernel alpha. And then there's a generalization of algebraic relation is lambda plus this overall degradation rate is equal to the catalytic spectrum at this growth rate. And I think this is a new formula that I haven't seen this in like mathematical biology or physics books, because this is derived from the DDE, which is also not the traditional DDE on the textbook. So I think this is relatively new. So thank you, and I want to emphasize that traditionally this type of DDE, Volterra integral equation, has been studied for 100 years. But the DDE we mentioned today is less studied. So I think in the future it will be interesting to know more about this type of DDE, which is you can think this is like second derivative of the DDE, yeah, because this dz over dt and this is the integral, yeah. And thank you.
Thank you, Dr. Lin. So, giving us a very interesting deep talk. So, any questions, comments, remarks, or counter examples from the audience? Yeah, please, Professor Wang.
就是说 x 这些 coefficient， 它全部都会 summarize 在 alpha、beta、gamma 里面。那就是 OK， 所以您的问题是说，如果我不解 ODE， 我怎么做 ergodic average， 对不对？所以实际上，如果它不是 fixed point， 我还是需要知道它的。对 ，sorry， 这个 z 的 simplex dynamics 不一定跟 x 的是一样的，因为 z 这个是 只有一个 variable 嘛，所以它等于就是它就是一个 variable 而已，它没有 simplex， 没有办法 ，simplex 还是要从原本 ODE 解。对，第一个就是 alpha， 就是这个微烃碳其实是可以量的，就是可以用 radioisotope 来，你可以喂这个细胞葡萄糖 label 的，然后看它多久，最后这些葡萄糖被做成 transporter， 所以这是本身有一个自催化上的物理意义，生物物理上的意义。那 beta 就是那个 transporter overall degradation。那另外一种 perspective 就是说，为什么要做这种 phenological 的 alpha beta， 而不是做每一个 detail 的 kinetics？ 因为实际上我们没有办法知道 detail kinetics， 太复杂。但我们可以 phenologically 去大约近似实际上的细胞的 alpha beta 大约是长怎么样子。对，所以这个是这个 approach 可以帮助的地方。alpha beta 可以如果我们真的知道每一个 detail flux， 我们可以去算 alpha beta。那就像我刚刚讲，就是要找这个 first heating pathway 去一条一条的把它加起来，就会得到这个 alpha。那贝塔有另外一个公式，所以知道 exactly 的话，我们是可以算这个 alpha 跟贝塔。不知道原来 system 的话，可能要估计，就是说它的微烃碳是多少，就变成一种比较 hand waving， 就是说做实验上我每5分钟量一次，然后去算它的 isotope 什么时候会到达，这样子，就是实验上可以测，但是就变成就不是从这个 system 算。就是 system 如果它的 topology 很好，例如说一个 line， 它就算是在大 n， 我们也可以 analytic 算这个 alpha beta。那如果比较复杂的话，就变成它本身也比较复杂，对，但其实是可以。对对，那应该说，当一个系统很复杂的时候，不管是用矩阵算，还是用这个 kernel 算，都很难。但是 kernel 比较容易去估计它的 alpha 跟 beta。矩阵的 eigenvalue， 它的 eigenvalue 是蛮难从它的 coefficient 去估计一个值。就是说当然也是可以估计，但是对对，以数值上，我完全同意这一点，就是说数值上这一套不会比较简单，但是有一些棒，就像我刚刚提到这个。
我可以帮你补充一下。
对。
因为你如果是一个非线性系统，你算出所谓 eigenvalue 也是做线性化嘛，对不对 ？eigenvalue 它只能做到局部的指数成长。
对。
可是你要问 global 的指数成长，你没有办法用矩阵的方式来弄，可是好像可以用你这滴滴的方式来算那个。
对，就是如果它是非线性，先讲这个非线性好。假设它是。
符合你的假设的非线性。
对对对。假设是 scalable 假设它 converge 到一个 limit cycle 好了，那这个 limit cycle 的 long term growth rate 没有办法用矩阵算，但它可以用用这个 ergodic average 直接 numerical 算，或者是在那个 limit cycle 里面去算它的 alpha beta， 做一个 proper 的 average。那对，
所以我觉得在线性的世界里，这两个没有差很多，说明本来的 OD 的方法是不错。
对对。
可是非线性的世界里，或许你的方法就有一个
对。
我
觉得这个优势应该是更 phenological， 就是说，像这个例子好了，这是一个 n 维的矩阵，那它的 eigenvalue 我可以 analytic 算，但它可能 n 很大的时候没有 closed form， 但是它其实可以有一个 upper bound 跟一个 lower bound， 那这个 upper lower bound 怎么算出来，就是当然线性代数很好的，我想大家可能可以找出这种2×2的矩阵来作为它 upper 跟 lower， 但是这是另外一个方法，可以很快的看这个 spectrum 去 bound 它这个的话，这是另外一种 approach， 对。
remarks? Maybe I want to compare one thing. Can you
yes.
go to the slide of your DD and OD comparison? Like 就是有 OD
yeah,
and DD 的。I want to make one comment and I want to make sure whether it's correct or not. Maybe 在最后啊。Okay.
Yeah.
So before you have a nonlinear OD,
Yeah.
a lot of OD.
Yeah.
And now if you just want to calculate the upper exponent lambda,
Yeah.
then it's equivalent to study the linear DD.
Yeah.
Linear.
Yes.
Nonlinear or linear? Linear.
Okay.
Okay, suppose I don't care about this gamma. It's a linear OD, right?
This linear DD, but this alpha is after certain nonlinear average, right?
Yes.
Yeah.
So alpha does not, I mean,
Yeah. And once you get alpha, it behaves like a linear. Yeah.
so this is a surprising reduction, right? Nonlinear. Although you increase the dimensionality from
Yeah.
n to
Infinity. Okay.
but now you have a linear system. And
And I think the linear is because the scalable one, if you factor out this z, the remaining thing, you can do the average to make it. Yeah.
and another question is, where do you use a Markov assumption to derive this?
Oh, so in order to know the waiting time, need to have Markov assumption because let me go to the general.
If I don't have a Markov assumption, then the DD is not linear anymore. Can I say this? Or there's no DD reduction?
I think because Markov assumption, you can write the transition probability from one node to the next node by this formula. And this is linear. Okay, so this has only RT and it's proportional with U. But if no Markov, you can still have DD, but it's not linear. Yeah.
Oh, any other questions? So if not, then we should end the colloquium today here. Okay,